Kathryn Styles. I'm from the University of Warwick, and I'm working with Antonia Sagona, and we're specifically looking at phage resistance and specifically looking at transposable insertion elements and how that changes phage resistance. So this has been a 18-month project that I've been working on, so potentially have much less extensive data than some of the other speakers that have been through. Okay, so we're specifically studying pathogenic E. coli. I'll just move this bar across because I don't think you need to see that. And we're looking at this because E. coli, amongst other things, is one of the causative organisms associated with meningitis. And that's something that's particularly prevalent in neonates and young children. And specifically, this pathogenicity has been linked with strains that express a K1 capsule. So that's something made of polysialic acid. And that's something that allows the pathogen to evade the immune system due to similarities in structure to things that are found in the human. And it also allows it to cross the blood-brain barrier. So to target this, we're particularly interested in okay, wait, let me change my slides now. There we go. So we're specifically interested in K1 F bacteriophage, and that was first isolated back in the 1980s, and that's a T7-like bacteriophage. But instead of like T7, instead of having the T7 tail fiber gene, it's got a gene that instead encodes for an endosialidase, and that allows recognition and degradation of this polysialic acid capsule. So unlike T7, therefore, it's specific to, on the whole, pathogenic hosts, whereas T7 has been shown to be blocked by this capsule. And we've been studying this bacteriophage. I think just to make things a little bit easier in the lab, we've been using a model E. coli called EV36. And this is a hybrid of K12 and K1. So it's a non-pathogenic lab strain of E. coli, but it does express this K1 capsule because that's the bit we're interested in. So it did allow a bit more sort of flexibility in terms of what areas of the lab we were allowed into and the assays we could do. So just a bit of background as to where we went with this project. When we did so this project is in collaboration with the University of Edinburgh, and there are some aspects of it that are related to mathematical modelling. But when we started running our preliminary trials, we did notice that there was a lot of resistance that would develop quite quickly to K1F. So the project sort of went in that direction a lot more. And then we wanted to kind of understand how this resistance came about, because obviously this is very relevant to phage therapies, and then see obviously what the mechanisms exactly were in the hope obviously we could try and solve this in the future. So our main findings from this were that there was transposable element insertions that happen very quickly when you expose the E. coli to the K1F plasmid, bacteriophage, and you'd get these insertions specifically into the polysialic acid biosynthetic cluster, and that would then cause the resistance to K1F, which was obviously targeting this particular capsule. So the first thing we did was like a preliminary sort of screen. We exposed our K1 expressing strain to the bacteriophage, and you can see here on the image on the left, on the whole with K1F exposure, you get a large amount of clearance in a plaque assay, whereas for T7, which is largely blocked by the capsule, you get a small amount of clearance, which is potentially due to if there was, gaps in the polysialic acid, but on the whole, T7 doesn't use EV36 as a host, so you get this very small clearance. Whereas we found if we exposed our strain to the bacteriophage and then collected isolates after a sort of a longer period of time, and then profiled the resistant versus the non-resistant colonies, you would end up with, they would gain a susceptibility to T7, but then they'd lose their susceptibility to K1F and they become fully resistant. So this is something that we then wanted to sort of study further and see, have a look at a lot more of the isolates. So specifically, when we were doing these screens, we selected two mutants. We've labeled these just as mutant 1 and mutant 5, and we found that mutant 1, you got absolutely no clearance at all from K1F, so that seemed to have like full resistance. Whereas mutant 5, if you look back at the image that we get here, you'd get a tiny amount of little spotting here for the K1F. So it seemed like there was still a tiny amount of kill that was going on, but not nearly to the extent there was for the wild type. So we selected both those two specifically, and then we did sort of more screening on those. We did a lot more growth curves, some microscope work, things like that, to kind of assess what was going on with those. So we did a number of growth curves. These are for our negative controls. There's no bacteriophage here at all. We were just looking at relative growth rates, and we found that for both the mutants that showed resistance to our phage, we did get like there was a lower amount of growth during the time period. And I've also added in MG1655 as a control here, which is just another K1 strain that doesn't express the K1 capsule. So just to see if it's the capsule that's causing the better growth in EV36. So yeah, there was a fitness cost to both our mutants. And then when we actually start adding our bacteriophages in, so I'll try and move this so you can actually read what it says. For EV36, so that's shown in green. So this is what I was talking about, where you get a lot of resistance develops to K1F. So the green line is where we're exposing our wild type strain that's never seen a phage before to the K1F. And you see that you do get initial kill after we add the phage at 4 hours, and you do get initial kill up until about 8 hours, and then you get massive amounts of resistance that seem to develop to this bacteriophage. Then when we added the K1F to our mutant 1, so the one that appeared to be fully resistant from the plaque assays, you get very similar growth curves for both in the presence and absence of the K1F. So that did indeed seem to be resistant throughout. So that's the yellow and the light blue lines. Whereas for mutant 5, so that was the one that did seem to have some spotting on the plaque assay, and so therefore some susceptibility to K1F. That's the dark blue line. So this one that you can see here. And that did seem to be killed a small amount, but later, but then it would never develop resistance in quite the same way as the wild type one had. So you still get lower growth by the end of your time point, but then you're also not getting, you do still have growth. So there is some kind of resistance. And following on from that, we wanted to establish what happens with different bacteriophages. Sorry, I'm having issues with it changing my slide again. There we go. So we also decided to add T7. So as I mentioned previously, T7 is normally blocked by the K1 capsule. And so we wanted to see what would happen if we added, now that we know our cells are resistant to K1F, what would happen. And both of them, both mutant 1 and 5, did develop susceptibility to T7. But interestingly, they'd also start to develop some resistance as well during the 24-hour period. So this is our strains that have been exposed to K1F and now are sequentially being exposed to T7. So there should be no K1F left in the solution. And then when we added combinations of phages at the same time, you would get like very low growth rates, and you would see very little development of resistance. So following on from this, we wanted to understand if we were to, with our mutants that have been exposed to K1F, if we then expose them to T7, what would happen to the resistances? Are any of them reversible? Would we end up with going back to K1F susceptibility, or would they become resistant to both K1F and T7? So we did a number of more studies with these sequential mutants, where we'd exposed it first to K1F, selected the isolates mutant 1 and 5, and then exposed them freshly to T7, and then collected further isolates from these. And I do have the full growth curves for these, obviously, if anyone did want to see them, but just from the point of view of a lot of very chaotic lines on a graph and time in this presentation, I've just summarized this all as the relative bacterial growth rates, which we took at 6 hours. So this is during the middle of the exponential phase in this study. And the ones that are of particular note is that for mutant 5A, so this is one of the sequential mutants, we got double resistance. So it was resistant to both K1F and T7 by the end. So it seems to have got at least, I guess, 2 changes to its genome. whereas mutant 5B was the relative growth rate was statistically similar to the one that we got for the wild type, and it gave very similar growth curves again to the wild type. So it was susceptible, but then seemed to develop resistance over the 24 hours to K1F, and then resistant, but seemed slightly susceptible by the end to T7. So that strain there in particular seems almost to have reverted back. So it indicated it was a reversible change that was causing these mutations. So obviously it's something we wanted to prove and find out further. So the first thing we did was we got some confocal microscope images. We wanted to see what exactly was happening to this K1 capsule. And so on the left, as you can see, there's EV36, that's the wild type strain. We've highlighted so DNA is in blue, and then we've got in red is an antibody that's specific to the K1 capsule. So you can see there nicely the capsules around the entire cell. And then for mutant 1, which was the one that was fully resistant, you see in a lot of the cells, you'd get this clustering of the polysialic acid at one end or the other of the cell. And then for mutant 5, which was the semi-resistant one, you'd see absolutely no capsule at all. But then for mutant 1A, which was sequential from mutant 1, so like its offspring, you then get a return of the polysialic acid being produced, as you can see with the capsule there. Whereas for mutant 5A, you would get it would remain the same. There was no polysialic acid. So following on from this, we wanted to understand the genetics, obviously, behind this to prove that our ideas that it was reversible were correct, things like that. So the polysialic acid biosynthetic gene cluster is a 17 kilobase biosynthetic cluster. It's split into 3 regions, but 2 of them, region 1 and 3, are both the KPS genes, and they're involved in assembly and transport of the polysialic acid. And region 2 is the one that's involved in biosynthesis, activation and polymerization of the polysialic acid. And we were expecting that there would potentially be a change to somewhere in this. And so mutants 1 and 5 initially were sent for long read sequencing to see what happened. And it came back, the rest of the genome was entirely intact. Everything was as we expected. It came back with some very lovely contigs. But there was very little question, like the reads were very long, and we found that in mutant 1 there was a disruption to the KPSE gene, and so that's one involved in polysialic acid transport. And in mutant 5 it was a disruption to the NEUC gene, which is involved in biosynthesis. So just to cross-reference that with what we saw for the microscope images. So for mutant 1, that's the one that seems to be a mutant in transport. This would make sense. It's still fully making the polysialic acid, but then it's taking it to the poles of the cell, or sometimes we find it sort of clustered throughout the cell. And then for mutant 5, this is the one that was defective in the actual synthesis. As you'd expect, you get absolutely no polysialic acid. And just to quickly check this as well, we did a rescue experiment. We added the NEUC back into our mutant 5, and we did get a return of the phenotype of polysialic acid synthesis return for that one. So that's what we did there. And then we did also do some short read sequencing of some of the other mutants, but because of the way the transposable element insertions went into the genome, we found that with short read sequencing it wasn't nearly as clear. They would end up sort of splitting contigs, and so if we want to properly analyze what exactly has happened with all the other mutants, we'll need to do long read sequencing, which is obviously a lot more costly and takes quite a lot more time. But from what we could see from the other mutants, the ones that then started expressing the polysialic acid genes again seem to have the phenotype of that. They seem to again obviously have the intact biosynthetic cluster. So it does seem to be that this resistance mechanism is reversible. But the resistance also always seemed to come at a fitness cost. The cells never seemed to grow as well in growth curves that we did. And they also obviously resulted in susceptibility to T7 because the capsule was then also gone. So following on from this, we're hoping we're going to get some long read sequencing done of some of the other mutants. There's a few more recovery experiments we want to do. And hopefully, obviously, if we get more funding for the project, that would allow for some of these things to be done in a lot more detail as well. But this research has now all gone to a paper, and yeah, so we're just doing our last sort of checks through that, and hopefully that'll be going into review, and you'll be seeing it before the end of the year. So yeah, thank you to everyone in the Sagona group for all their help, and Aidan, who's my co-supervisor over at the University of Edinburgh, and also Rebecca and Lauren at the University of Bath, who've been helping me with analysing the genome sequences, and also EPSRC funding the project. Thank you.